welcome back to our channel. I'm back with yet another video. This time I'm going to talk about the regulation of glycolysis. Glycolysis is an anaerobic process, which means it do not require oxygen to proceed and produces minimal amount of ATP. In our previous video, we have already talked about the detailed pathway of glycolysis and what enzymes are involved in this process. In this video, I'm going to talk about how this pathway is regulated. In fact, this pathway is regulated by regulating those enzymes which are involved in this pathway. Matter of fact, only 3 enzymes are actually involved in the regulation of glycolysis. What are those enzymes? First one is hexokinase, which catalyzes the first irreversible step of glycolysis. And the second enzyme is Phosphofructokinase 1, which is inhibited by its own product, that is fructose 1,6-bisphosphate. Plus, it is also inhibited by ATP and citrate, which is coming from TCA cycle. Again, phosphofructokinase is also activated by some of the agents. Which are those agents? That is ATP, ADP, AMP and fructose 2,6-bisphosphate. We will talk about fructose 2,6-bisphosphate later in the video. The next enzyme that is regulating the glycolysis is pyruvate kinase. Pyruvate kinase is inhibited by ATP, aniline and acetyl-CoA, and also it is activated by fructose 1,6-bisphosphate and phosphoenol pyruvate. Now, how these enzymes are activated or inhibited by these substances or agent, we will talk about this in detail in this video. So keep watching. As discussed in the initial segments, the regulation of glycolysis occurs by controlling 3 enzymes that catalyze spontaneous reactions. The enzymes are hexokinase, phosphofructokinase and pyruvate kinase. The regulation system of glycolysis actually occurs by 2 ways. One, by local control, that depends upon the concentration of the pathway substrates and intermediates, and this local control helps the cell in which the glycolysis is occurring. And the second system is global control, that involves hormone activated signal cascades, and this system benefits the whole organism, that is the entire body. Hexokinase controls the first irreversible step of glycolysis and converts glucose to glucose 6-phosphate. Glucose enters the cell through the glucose receptor, and also when the cell needs glucose, exit the cell via the same receptor. Cells simply trap the glucose by phosphorylating it and prevents its exit through the glucose carrier. Hexokinase is inhibited by its own product, that is glucose 6-phosphate. Product inhibition of hexokinase ensures that cell will not continue to accumulate glucose from the blood if glucose 6-phosphate within the cell is ample. Now let's talk about the different forms of hexokinase. Hexokinase, we all know, have 4 forms. Hexokinase 1, 2, 3 and 4. Hexokinase 4 is called glucokinase. Hexokinase 4 or glucokinase, which is found in the liver, has high Km, thus activated at high glucose level. Now what is this Km? Km denotes the enzyme's affinity towards substrate and is inversely related with the affinity, which means the more the Km, less the enzyme's affinity towards substrate. As it is mentioned that hexokinase 4 has high Km, it means it will activate at high glucose level. One effect of insulin is that when high blood glucose level occurs mainly after the feeding state, the insulin is released from the pancreatic beta cell, which binds to its receptor, that is receptor tyrosine kinase, which is found on the liver. And in the hepatocyte, it activates a gene that is called glucokinase genes, and glucokinase enzyme is produced. glucokinase is not subjected towards the glucose 6-phosphate inhibition. Thus, liver will take up and phosphorylate glucose even when glucose 6-phosphate is high inside the cell. The fate of glucose 6-phosphate inside the liver is shown in the diagram. It can move through the glycolytic pathway and produce pyruvate, or it can move to the glycogen synthesis pathway and glycogen is produced, as glycogen is the storage form of glucose. In the liver, the excess glucose 6-phosphate is usually synthesized to glycogen. Another important enzyme which is present in the liver is glucose 6-phosphatase, which hydrolyzes glucose 6-phosphate and produces glucose. Its function we will discuss in some other videos. In my previous video, I have already talked about that glucokinase is activated at much high blood glucose level in compared to hexokinase 1, 2, 3. In this diagram, it is clearly visible that hexokinase 4, that is glucokinase, is activating at very high glucose concentration in compared to hexokinase 1, which is activating at very low glucose concentration. Now let's move to the next enzyme, that is phosphofructokinase. There are actually two phosphofructokinase isozymes. Phosphofructokinase 1, that produces fructose 1,6-bisphosphate. The other one is phosphofructokinase 2, that produces fructose 2,6-bisphosphate, which is a potent allosteric regulator of the PFK1 activity. Phosphofructokinase is allosterically inhibited by ATP. Now, what is this allosteric enzyme? We all know that enzyme has an active site in which substrate bind to it and gets converted into product. Allosteric enzymes has another site where the regulators binds to it and controls the enzyme's activity. Either it can enhance the enzyme's activity or it can decreases the enzyme's activity. Those sites other than the active site are called allosteric site, and the enzyme that possesses those sites are called allosteric enzymes. PFK exists in two conformations, that is one R state and T state. The R state is thermodynamically stable, and in this state enzyme works at its optimum capacity, and this T state is not thermodynamically stable, and thus why they do not perform at their optimum level or become inhibited in the T state. We see that the PFK is allosterically inhibited by ATP. When ATP is at low level, ATP level is low, the enzyme assumes the R state and function at optimum level. But when ATP concentration is high, ATP binds to the low affinity regulatory site and promotes the tense conformation, that is T state. Here the activity of PFK is shown in different concentration of ATP. At high concentration of ATP, we get a sigmoidal curve, that is the activity of the enzyme is decreased. But at low ATP conformation, we get the hyperbolic curve, where the activity of the enzyme is maximal. In this diagram, it is shown that when the ATP level is high, the ATP is bound to all its regulatory region and causes change in the substrate binding site of the enzyme. That's why the substrate cannot bind to the enzyme properly, and thus the enzyme become inactivated at T state. But when ATP is low in the cell, invariably the AMP or ADP concentration will increase. That causes the also change in the active side of the enzyme, and the enzyme assumes a perfect shape, that is perfect conformation, so that the substrate can bind to its active site, and thus at R state the enzyme became active. So when ATP level is high, the enzyme assumes T state, decreases fructose 6-phosphate binding to the catalytic site, just like I have said. When ATP is low, that is when AMP level is high, the enzyme assumes R state, increases fructose 6-phosphate binding to the. In this graph, the activity of the PFK is shown. PFK is least active when the ATP concentration in the cell is high, and the activity of PFK is highest when fructose 2,6 bisphosphate is present in the cell. Fructose 2,6 bisphosphate was produced when the enzyme PFK2 was active. So AMP, ADP and fructose 2,6 bisphosphate are the activators of PFK1 activity, and ATP and citrate functions as inhibitors. The last enzyme that controls the glycolysis process is pyruvate kinase. It converts phosphoenol pyruvate to pyruvate. It is the last step of glycolysis and is controlled in the liver partly by modulation of amount of enzyme. High glucose within liver cell causes transcription factor, carbohydrate response element binding protein to be translocated into the nucleus, where it activates transcription of the gene pyruvate kinase. This facilitates converting the excess glucose to pyruvate, which is metabolized to the acetyl-CoA, the main precursor for synthesis of fatty acid for long-term energy stores. What other agents that control the pyruvate kinase? Pyruvate kinase is negatively regulated by ATP, alanine, acetyl-CoA and cyclic AMP dependent phosphorylation, and is positively regulated by fructose 1,6 bisphosphate. So this is it guys, that's all what I have for today. If you have any question, do comment on the comment section, and if you like the video, do not forget to hit the like button and do subscribe to our channel for more such videos. Thank you.